I'm Roger Hanlon, a senior scientist at the Marine Biological Laboratory in Woods Hole. In part one of this series, I talked about some of the concepts and general questions about this subject, signaling and camouflage in cephalopods, and laid the groundwork for how this unique capability occurs. In part two, I talked in more detail about the visual control of the multiple patterns that the animals can deploy. And here in part three, I'm going to talk about the skin of these animals that allows the amazing visual diversity that they can produce. I will give some small hints as well about there are some folks in the material science and engineering communities and the nanotechnology world who are beginning to look at animal systems for some bio-inspired approaches to new kinds of adaptable materials. So to begin with, you can see right here in the image, I've got some of the typical chromatophore organs in the skin of a cephalopod. These are pigmented organs that I'll describe in detail momentarily. But here you see this image of blue iridescence and the exact same piece of skin right there showing not blue but sort of a pinkish red iridescence. Now this kind of iridescence, this is structural coloration. That means that unlike the pigments, which absorb some light and reflect others, structural coloration devices absorb nothing. They purely reflect light. And in many cases, it's very angle dependent. So what's happening here between the blue and the red is that the light has been shifted in direction to give a different coloration. So these are very different ways of handling light, but I want to make the point right from the beginning that the optical diversity shown by these animals is largely enabled by having structural coloration and pigmentary coloration in multiple layers of the skin. And this is a different way to approach creating materials or anything that has coloration and patterning, and the animal kingdom does a lot of this. So maybe there's something biology can contribute even to our industrial world. So without further ado, I want to go and remind ourselves that we're looking at animal patterning. This is the giant Australian cuttlefish. It's not quite as big as that relative to me, but it does get up to about this big. They look like little submarines, very beautiful animals. And this large animal swimming right there can, within a second or two, just descend on the bottom and disappear like this even when you full frame it. So the details of this patterning are very interesting. I covered some of those in part two, but I want to talk about right now are the details of the light and dark bits and how they create these different appearances. So, first of all, there are millions of pigment organs, the chromatophores, that are creating this pattern, but there is some iridescence and there's all that whiteness in the skin as well. Where does white come from? White is actually a unification, if you will, of all wavelengths together. That's what white is. So to create white in the skin means that the animal has to collate all of the available wavelengths in the vicinity and then distribute them out in all directions as white. And we're going to show you how this animal does that. Now, I've already mentioned that there are two systems here. One is the pigmentary color shown in the squid. You can see a lot of the patterning here and elsewhere. These are what we call long wavelengths, your reds and your yellows. It's one side of the spectrum. But how do you get the short end of the spectrum? By the way, these are under active control. I'm going to show you a lot of that in a minute, which does mean that all of these pigment organs are controlled directly from the brain and to create the patterning. Now, in addition, you have this very important set of structural coloration devices. These create the complementary end of the visible spectrum, the short wavelengths, the blues and the greens and others, and they create some of the white here, depending on what it is. So some of these are actively controlled, but many of them are just sitting in the skin doing their trick with light. And so some are actively controlled, some are passive, and both have certain features and advantages or disadvantages. Okay, now this is from a NOVA special in which you can see the yellow chromatophore, so it's a tight pigment sac. These radial muscles attach to the periphery, and when the muscle contracts, it pulls this out into a disc of color. And when there's no neural signal to the muscle relaxes, and boink, it goes back in, and you don't see that little point of color. So you can see the red, the brown, and the yellow chromatophores. Now we go under the skin. There are 3 layers of chromatophores, and you see this iridescent pink and green and blue, and then your white base layer down here. So collectively, you've got the full spectrum of color right here in this layered skin. And this is not unlike the artist's palette, except that most artists are using mainly pigments. Others use reflectors. These animals are using both. All right, so we can measure the colors in the skin very carefully with a spectrometer. Here's an example of some chromatophores in the skin. If you look where the arrow is right here, you see a yellow chromatophore, and it has opened on top of some blue iridescence. So over here we can measure the chromatophore, red chromatophore here, the yellow chromatophore there, or sorry, this is the blue iridescence, and when it's over the top, this is the measurement you get. So you get an intermediate color because the light is going through a pigment, and it's also reflecting off a reflector, and it's coming back through the pigment, and that's different from the two independently. So this is part of the complication, or part of the interesting part of the skin. Here's another example with some different colors, but you can see that we can measure these with precision to see what the interaction is between pigments and reflectors. So the control of the animal is interesting. Here's a cutaway right here of a squid head. You know, the eye would be right here, but if you remove the eye, you've got a brain. And this number 1 right here is the optic lobe. The optic lobes are enormous in this animal. It's a very visual animal. And the optic lobes send information down to the main part of the brain. Number 2, the set of lobes called the lateral basal lobes, they integrate the information from the left and right eye, and they send information to the lower motor center in the brain. Right here, number 3, it's called the posterior chromatophore lobe because it operates all the chromatophores on the posterior part of the body, and there's an anterior chromatophore lobe here in number 3. It operates the millions of chromatophores on the arms. So you have a hierarchy of control from the optic lobe to an intermediate brain center coordinating that information and telling the lower motor centers how to control each of the millions of chromatophores that are out there in the periphery. So yes, this is expensive skin in the sense that it's highly innervated, it's complicated. But it gives direct active control of all those little dots in the skin. And from those multiple dots, you create patterning. And let me show you a little bit about how that works. So here we got an example of a piece of skin on a cuttlefish. And this is motor neuron right here that's controlling the chromatophores here, and another motor neuron from the brain that controls all of these chromatophores. When the signal comes from the brain to turn those on, you get this sort of patterning right here. Now there's an equal number of chromatophores all the way through here. It's just that these are expressed, these are not expressed. So it's an on or off kind of system, and they can turn it on to a greater dimension of the chromatophore or a lesser dimension to make this line darker or lighter. I also point out that the white business underneath, and you can see it in the whole animal right here, has those white reflectors concentrated here so that they can create a high contrast zebra pattern for signaling. So the skin is constructed in a very specific way. But that area right there that are just highlighted are areas here where no neural signal's said to turn on, so they're just lying there, and there's so little you don't see them on a gross scale. Now here's an example of a different part. Right here you see this spot right here, and you can see it expressed down here in the animal, and up here it's not expressed. And the point is that each of these chromatophores can be expressed in different parts of the pattern. So there's very intricate control. Some can be combined with others or turned on or turned off in a rather complicated matrix. And then the other thing that can happen is this. I'm going to show a video of this in just a moment, but to set the stage, I have a stimulating electrode here, and there's the same stimulating electrode. And we're looking down in the skin here and putting the electrode directly on one of these white spots. And down here, I'm just moving the electrode another millimeter or 2 over here, and in this case, as I move the electrode from here to there, you see that the chromatophores in this area, they surround the white bits to enhance them, and up here, when you stimulate on there, they mask the adjacent white markings. So by this simple movement of the stimulating electrode on top of a white spot or to the side of it, you can actually see the distribution of the motor neurons and which pigment chromatophores they control. So if we look at this in a dynamic sense, here's one of those white spots. So I'm going to be moving the electrode from here to there, to there, to there in the following video. And now you're going to see, now I'm here, now I'm there. Now I'm here, now I'm there. And now you can begin to see how it works, and you also get this sort of plain low contrast look, a bit like this, and a high contrast look a moment ago when it's in between. So again, by a somewhat simple mechanism of having the skin structured the right way and having the control over it, you can take advantage of these passive white markings, and you can use the very active pigmentary color to complement it. It's really very beautiful as well. So what's a chromatophore? A chromatophore is some kind of pigment cell. They occur in many animals, but I want to point out that they're totally different structures, and the word chromatophore is not appropriate because it means something very different in a vertebrate versus an invertebrate. In fish, you have a fixed cell type. The cell shape never changes. What changes is the distribution. Either the pigment granules are condensed in one place, or they're distributed throughout the fixed cell. Down here in a frog or a lizard, you have the same thing, a fixed cell. The pigments are either in the bottom and you don't see them, or the pigments migrate up here and you do see them. All right? Down here, it's completely different. As I showed you in the animation, you've got a tiny little pigment chamber right there, and then the muscles pull it out into a disc of color. Super active kind of process. Okay, so the first two, well, the first one can be, this one can be a little bit fast in fishes, sometimes very fast. This is always a slow process. And down here, when you have direct neuromuscular control, you have extremely fast, fine-tuned precision. Here's what some of those chromatophores look like. You've seen a few of them. Some are red, yellow, red, and brown are the predominant colors. They can go sort of pinkish in different species right there. And now that organ, and I emphasize this is an organ. There are several cell types here. This is not a single cell. So at the beginning, you've got pigment granules right here in the middle. You've got radial muscles down here. There are 18 to 20 radial muscles to pull this out. You've got nerve fibers right here that actuate each one of the muscles. And then you've got this clever thing here called a cytoelastic sacculus, and that's got a stretchiness that closes the chromatophore when the muscles react. So this is a complicated organ. We don't know as many details about how it works as we'd like, but we are studying this. But here it is in action. You can see this chromatophore right here, this brown one, opening and closing. And you can see the muscles down in here pulling it out. And so there's a yellow chromatophore right there that's opening and closing. This is a preparation, a fixed prep. This is not in the live animal, but you get the idea of how it works. And so it's pulling in all directions. And if you think about the skin, there are muscles overlapping all over the place. So it gets kind of crowded for the muscles even in that skin. Here I'm going to show a lot of patterning in the skin. And now this is a live squid. There's no anesthesia or anything. The animal's sitting down there very happily. And we're going to use a high-definition video camera to zoom closer and closer into the skin so you can begin to see a little bit about how it works. So as we go down in the skin, these chromatophores are expanded. These are not. You can see some of the underlying iridescence. And as we move, posteriorly to down the animal, you can see where you can create these transverse bars. I want to point out that all of these chromatophores are actually moving a little bit. They're not actually static all the time, because on the gross scale, you don't see that, and the nerves don't seem to operate that well. I'm going to speed this up a little bit just so you can see a little more of what's going on. We'll go down the skin a little bit. Now we're going about double speed. All that movement in the background is food thrashing around in the stomach. The animals are translucent. But now you see this bit of patterning right here, and as we zoom in, we begin to see the real details of that patterning. Now watch carefully. In this image, we've gone from patterning and the animal closes some more, and suddenly it closes all of them. So we've created that patterning by opening some and closing others. Very fine precision, very beautiful skin. Now this is an example right here of squid skin. And the squid skin is what we kind of glibly call low resolution skin. It's kind of like a computer monitor that doesn't have a high resolution, which is to say that the chromatophores, whether they're closed or whether they're open, are far apart and they're very big and they're not very dense per unit area. So this is a drawing right here of the other two. Down here you have a cuttlefish with the same scale bar, and here in the cuttlefish you have hundreds more chromatophores per square millimeter than you do in the case of the squid. So you can have sort of gross patterning or you can have very fine detailed patterning depending on how big your individual chromatophores are. The structural coloration cells, the iridophores, this is just an example, 4 examples of what the iridescence could look like. But I did point out earlier that some of these are passive cells just sitting there, but a few of the structural coloration items like the iridescence are active. And here's an example, I'll get out of the way here, of a squid underwater that I filmed. Look at this pink iridescence on the ventral part of the animal right under here. This is all under control of the animal, so they can create this iridescence for some specific uses. But here you can see how delicate that skin is and how beautiful that iridescence is. Now, how did these things work? I can show you this just in 2 slides. Essentially, here's a piece of skin in the non-iridescent state. Here is the exact same piece of skin in the iridescent state. Between here and here, we added the neurotransmitter acetylcholine, which also occurs in your body as well. And the acetylcholine created iridescence in these cells. And if we look at the electron micrograph, here is the case when it's not iridescent. And whatever this material is in this platelet, it's dispersed and the light's going straight through it, all the way through, boink. In this case, however, something has condensed. Now the light comes down, hits that platelet, and bounces off of it. And that's from the action of the acetylcholine. So we described that years ago. And we'll point out right here that this is all just below that platelet. You follow this line all the way out. That's extracellular space. It's actually connected out here. So the acetylcholine, which is a chemical, is actually getting dispersed throughout this skin, and then it diffuses down in between these cells, attaches right onto the cell wall, and changes the platelet to make it iridescent. So there are 2 mechanisms going on. The acetylcholine will turn the iridescence or the reflection on. And number 2, it can change the color by adding more acetylcholine, shown here. So in this case right here, this is the non-iridescent state right here. Now it becomes iridescent of a pink and gold, and when more and more acetylcholine is added, like here and in here, the colors go to a mix of colors, then they go to your shorter wavelength colors. And they do that by making the reflecting platelet goes thick, thinner, thinnest, and the other space goes just the opposite. So you have high refractive index, low refractive index, creating this kind of reflection. Now, the figure at the bottom shows the actual concentrations of acetylcholine for each of those 4 treatments. And you can see these are physiological quantities of acetylcholine, so it's a true physiological titer that we're showing here. There's the message. This is chemically tuned color for structural coloration. Very unusual mechanism. All right, what's really interesting here is that several years ago, Wendy Crooks and Margaret McFall and colleagues published a remarkable paper in Science looking at that material that's reflecting the light in the iridophore. And it turns out to be a protein, and it's an unusual protein that they named the reflectins, aptly named, I would say. But the animal world doesn't reflect light with protein. This is a very unusual thing. But this reflectant protein has some remarkable qualities, some of which are shown in this very nice paper published in Nature Materials by Rajesh Naik and Ryan Kramer and his colleagues, showing that if you take some of this reflectant protein and you make more of it in a laboratory and you spread it out on thin films, these individual protein molecules self-organize into a thin film and reflect light. And this is a really, truly remarkable capability. One could now even think about making some reflective material out of this protein. So it sounds very space age, as the old term goes, but this is a case where biotechnology is showing us some very interesting features. In this case, coming out of the so-called lowly squid, turning into a possibility of showing us how to make some very different materials. Ned Thomas and his group at MIT up in Cambridge, Massachusetts, have also used this idea of how iridophores work to create some of these different photonic gels. And they've published some of this information recently, shown down here, to where you can get some of the electrochemical color tuning. That's more or less what the squid is doing too. An electrochemical, electro being neuro, releasing some acetylcholine, creating a difference in the dimensions of these Bragg reflectors. So it's really starting to inspire the materials community, and I must say the materials community is asking the biologists some very tough questions about how it all works as well. Beautiful multidisciplinary enterprise. So a brief summary of the iridophore story is that some are active and some are passive. The active ones are acetylcholine. There are two mechanisms, one to become iridescent, the other is to change color, and that's chemically. And I hasten to point out that there are no muscles or nerves attached to these cells. So that's really unlike the chromatophores completely. And what happens is acetylcholine is changing the ultrastructure of this protein. It's changing its conformation. And one light goes through it, and in another, the light bounces off the individual molecules. So it's really happening at that molecular or nano scale. And the molecular switch that's going on in those cells is being worked out by Dan Morris at UC Santa Barbara and many of his colleagues as well. Again, this is thin film interference, but there are other optical mechanisms of how the reflection is actually produced. So just one quick slide to point out that what do we do with this information? The idea is that this information needs to be modeled according to the spectrometry and the ultrastructure of the skin. Here's one sample publication that we've done that we can look at the cephalopod skin and we can begin to demonstrate with some real metrics how the light is interacting with the pigments and the reflectors, and then you can go off and begin to make materials that may use some of those features. So the penultimate thing I want to look at are leucophores. Leucophore means white cell. I already mentioned these earlier. And these are assemblages of spheres mixed with the flat plate iridophores to create the whiteness. Now these are passive cells. There are no nerves, no muscles, no chemicals, no nothing. They're sitting there doing this handling of light to create white light in every direction. And we want to look, those are protein molecules too. We don't know exactly what they are, but we're beginning to look into that. So there's lots of whiteness in the skin. You can see this cuttlefish here has various kinds of whiteness. It's used to mitigate the shadows for camouflage. It creates maximal tonal contrast between the light and the dark bits for disruptive coloration, even create the optical illusion of three-dimensionality. That's called pictorial relief. And it can act as a white or light base layer upon which all the pigments are laid on top of, as I showed you in the video a few minutes ago. So here's a video scene of showing how white is important in signaling. Here's an octopus in the Caribbean. As I was filming it, it's showing me this remarkable display standing up on 2 of its 8 arms and making sure that I see the maximum whiteness and spread of the web and creating that high contrast whiteness with the dark chromatophores to give what we call a deimatic display. That means a threat or startle display. So white is very important as a signal as well because it creates that high contrast. If we look at a cuttlefish right here, there's the full cuttlefish, and we can go down to a piece of a fin where the white spots are. We look at that white spot in more detail. It looks like that. And then we can look at it in a light micrograph, and you can see some of these leucophores spread around in here. There's some iridophores as well. Now we can go down even greater detail down here to a transmission electron micrograph. We blow that up a little bit, and now you see these spherical leucosomes, and they have the adjacent flat iridophore platelets, and they're all co-located. And if you look very carefully now at some of these spheres, you look at them with a transmission electron micrograph right there, and you see all these little spheres right here. So all these little spheres are creating most of the whiteness, and we wanted to compare that whiteness in a cuttlefish with this white beetle, which is acknowledged to be able to produce some of the brightest white known in the animal kingdom. A beautiful paper published by Pete Vukusic at Exeter University in England in Science, and we decided to compare those. So Lydia Mathger in our lab did the spectrometry on the Leucophores, and here you see this flat line at high reflectance all the way across the visible spectrum. That's true whiteness, when every wavelength is equally shown at a high reflectance. This is really quite remarkable if you show it to an optical physicist. So, moreover, she did something very important right here. She took the light up and down or all the way around, and no matter which direction you looked at it here, you got this beautiful flat white line. So no matter what direction you look at, you're getting beautiful, perfect white. So here are some of the data comparing the beetle right here, that's the green line, with the cuttlefish, and this is over right here from 400 to roughly 700 or 800. That's the visible spectrum, and you can see throughout that period the lines are absolutely flat. Both of them have the same high degree of reflectance of pure white. However, one's a terrestrial animal working in the air, and the cuttlefish is a marine animal working in the water. So Lydia did the following experiment. She took the cuttlefish skin and she dried it, so got rid of all the moisture, desiccated it. And then she took the beetle and she wet it and got it all wet, and she did these tests again. And it turns out that the cuttlefish retains its same capability, wet or dry, to show whiteness in every direction, whereas the beetle lost all of its capability for distributing white light once it got wet. So they work by a totally different mechanism. Both are very efficient, but there's something unusually strange about the cuttlefish because it works wet or dry. So, I mean, this just opens the door for someone to look at this in a lot more detail. Now, here's a little detail for you, and you can see a scanning electron micrograph of one of those leucophores, and right here you see all these round spheres. You can see them in more detail, co-located with these flat platelets that are very characteristic of the iridophores. So this combination is very important, but we haven't figured out exactly how it works. Here is a close-up of some of the spheres that have the protein in them that create that whiteness. They're all tethered, we've just discovered, and so we're just beginning to try to understand what the protein is and what its light reflective capabilities are. And you can also look at these in a different way with some confocal microscopy. These are the iridophores right here, and here you have the leucophores right here. You can begin to see the three-dimensionality of it all. And here's a better representation of some of these spheres, the leucophore spheres, in the actual three-dimensional array. And this is very important for modeling to understand what those dimensions are, not only the various diameters of the round spheres, but how they're organized in 3 dimensions. So this work is proceeding now as well. So the skin papillae, here you see the cuttlefish with the smooth skin, and there come the papillae. You already saw that in part 2, but here's another example just to remind those who haven't reviewed part 2 that you've got this musculature that is very malleable, if you will, to cause 3D structure in the skin. So underwater, you can see here's a small octopus in California, and it's not showing camouflage, not showing very much papillae, but as it moves over and settles in the kelp, it suddenly looks like this. I don't know if you can see the octopus, but it's actually all of this is octopus right here. And you can see lots of these long tendrils of papillae. Of course, there are lots of tendrils of algae and everything else in the background, so this three-dimensionality not only obscures the edges of this animal, but it also blends in with all the other little tendrils. Now in the close-up, we can take the same species in the lab, and now we're looking at a very close-up of the skin, but watch very carefully about right there, and you can see one of those papillae jump up, and now in slow motion, it's going back into the skin, so you can see some of the kinetics. So now you see that this individual papilla was very, very well controlled. So we're beginning to look at how this shape deformation occurs, working with some engineers who can study that, and we're looking at the musculature and the basis of that as well. And of course, as I said in the earlier discussion in part 2, this is visually driven behavior, so controlled by the brain of the animal. All right, just one last thing is to look at an octopus. This is a small octopus that mimics flounders or flatfish in the Caribbean. Macrotitopus cephalopod is the name of this creature. It's got these extreme arms. And look at this beautiful fine papillae all over the skin. And again, they can control that to be perfectly smooth, or in this sandy habitat, you see the animal's all smooth right now, it can change quite a lot. So, in summary, to briefly state what we've talked about today, this transition from biology to inspired materials is well underway after a decade of discussion. There really are some animal models, this is just one example, that can inspire different ways to form coloration, patterning, even 3D texture. Some remarkable achievements have been made recently. I pointed out one of them with the thin films with the iridophores from the squid. And very different disciplines. This is a highly multidisciplinary enterprise. Biology with material scientists, optical engineers, and so forth can pose good questions to each other. It's really a stimulating way to look at some of these things. And I'll end this particular talk with Nobel laureate phrase, Albert Szent-Györgyi, discovery is to see what everyone has seen and to think what no one has thought. It's really been an inspiring idea for many generations of MBL scientists and others outside. And finally, again, my favorite saying that's in the library at the MBL by Louis Agassiz in the 1890s, study nature, not books. Certainly that's my excuse to get out in the world and go diving a lot. So thank you for your attention. Appreciate it. Good luck.